Okay, so hi everyone, I'm Guglielmo Persiani and I'm a PhD student in Brisbane University. So my PhD is about the study of the microbiology of the coal mine groundwater with the study here of the South Wales. And today I would like to present you some results regarding the use of the stable isotopes to detect and to a bit of understand the role of microorganisms in coal mine groundwater. So I would like to thank Cashew has financed the project, BGS and NIV to have supported the analysis of the isotopes, and the coal authorities to have supported all the field activities in South Wales. So abandoned flooded coal mines can provide heating and cooling sources for district heat schemes, where the low enthalpy energy contained into the mines usually is exploited using heat pumps. So this simple picture here is basically, it's basically showing a model example about how the mine water can be used. We can use the deeper mine workings, which are usually contains warmer water, and we can reinject back the thermally spent water into the more superficial mine workings. So actually, this is allowing us a water balance equal to 0. And so this is a model example, but there are other ways how to use the mine workings in the heat for heating purpose. So we will focus then on more on the geochemistry of the coal mine waters, which is actually required to properly run the heat pumps and the heating schemes. So coal mine drainages, as already has been said as well in the first presentation, is usually rich in iron, and the iron scaling process are very common and also well known as a potential problem. But as well, we can have other problems related to the use of the mine water. So we can have, for example, hydrogen sulfide, which is a very corrosive agent against metals, and we can have methane, which actually poses risk of explosion, and as well, in general, they interact with the heat and safety of the heat exchanger. So it should be pointed out that in shallow aquifers, where we have anoxic condition, the methane and the hydrogen sulfide usually are the product of the microbial activity. So actually, geomicrobiology may play a role in understanding the geochemistry of the coal mine groundwater. So we develop a very simple diagram, very simplified diagram, representing the potential microbial interaction involving the carbon and the sulfur cycle in groundwater, where both of those biogeochemical cycles are well related, and where in South Wales we are dealing with circumneutral mine drainages, where the pH is around 6 to 7.5. So the dissolved inorganic carbon, the DIC, is a proxy of the alkalinity, which is actually the parameter that buffer the water, keeping the pH on those values between 6 and 7. So our isotopes fingerprint approach try to answer those 3 questions. So basically, which are the source of carbon in subsurface? Which are the source of alkalinity and organic carbon? Is a geogenic source, so is a basically older than 50,000 years old, or we are dealing with some more modern sources, and as well on which carbon source the methane production rely on, and shall we consider the sulfate reduction as an ongoing process? Shall we consider it in understanding the geochemistry of the coal mine groundwaters? So very quickly, see the study here. We are in South Wales Coalfield, when all coal measured work are contained into the Westphalia stage of the Carboniferous. So in the west area of the coalfield, the coal seams worked are interbed with sandstone and siltstones, while when we move on to the east here of the coalfield, the coal seams worked are interbed with more shale and mudstones. So the site investigated are 4. They are 3 adits, and 1 Krummi navigation is a gravitary discharge directly from a flooded shaft. So, as I said before, we're dealing with circumlateral mine drainage, and where we have a variable iron content, where one site investigated here in Sarwet, which is classified as acid mine drainage due to the high iron content and a lower alkalinity values, while the other sites are all net alkaline with more reducing condition. So all the samples plot to the mean global meteoric water line. So means basically that the water is derived from recent meteoric infiltration, and we can exclude then a deeper and ancient groundwater interaction with our coal mine groundwaters. So here I'm showing the result of the radiocarbon content in the DIC and the carbon-13 in the DIC, plotted against each other, and as well plotted against the concentration, the inverse concentration of this solid anorganic carbon. So the lines that we see in those graphs are basically they're there to give us an idea about what would be the idealized mixed binary mixed model between a geogenic source and a model source, because those two sources, they have a very different fingerprint, both in carbon-13 and in radiocarbon. Okay, while the geogenic source usually has a carbon-13 equal to 0 and a radiocarbon value of 0, while the model source have a depleted values of carbon-13 and a idealized concentration of radiocarbon equal to 100. So as we can see here, the results of carbon-13 in our sites, they point out a clear interaction between a geogenic source and a model source, so where the carbonates and possibly the soil CO2, they are well mixed. But very interesting, with the increased value of the DIC, which is again a proxy of that activity, we find out that the content in radiocarbon increase as well, which is surprising and very interesting because, well, the source of alkalinity is believed to come from the carbonates, which are of course radiocarbon dead, and but those data suggest that we have another source of anorganic carbon where the alkalinity values are very high. So those results again are a bit interesting, as carbonate dissolution is assumed to be the main geochemical buffer in the coal mine groundwater, and well, if we assume that the carbonates are the main source of anorganic carbon, if we plot the ratio between the calcium and alkalinity, this ratio would be 1. And this is true for one site, which is also the net acidic site. While for the other 3 sites, we have a very lower ratio. And this can be explained with a cation exchange process, where actually we have a bind of the calcium and the release of sodium, okay, into the solution. But this again, if the cation exchange play a role in increasing the calcite solubility, this idealization of this process actually is not supported by radiocarbon data, because an increased calcite solubility would increase the DIC, but that DIC would have a concentration of radiocarbon very low, and this is not the case for our site. So now let's move on the characterization of the methane source for our site. So we find out that an high radiocarbon content in our sample usually corresponds to a biogenic signature, which is a very depleted signature of carbon-13 in methane. And again, also interesting, there is an apparent correlation between the radiocarbon content in methane and the DIC, which actually could suggest that the main biological pathway of methane generation, the biogenic methane generation, could be the carbon fixation and not the fermentation. So also to point out, where we have a biogenic source of methane, we find as well higher concentration of methane. Okay? And again, those higher concentration of methane rely on a model carbon source to actually generate that concentration. So then move on the stable isotopes analysis of the sulfate, where the isotopic composition of sulfate can suggest which reaction, one or two in the slide, could be primarily responsible for the pyrite oxidation. Actually, we are assuming that the pyrite is the only source of sulfate in our mine water, and this could be assumed as true for our study here in South Wales. So apparently, we have two sites where the isotopic fingerprint of oxygen in sulfate cannot really be explained by this binary mixing model, meaning that we have other processes that actually can alter this isotopic fingerprint in sulfate, which is, again, excluding the vaporites and seawater intrusion. We can assume that the sulfate reduction actually can enrich oxygen 18 in sulfate. So in those sites, sulfate reduction may take place. And when we look at the sulfur 34 fingerprint in sulfate, so I've plotted the data against literature data deriving from the analysis of the sulfur fingerprint from another coalfield, but the same geological time, just to give you an idea about which are the value of sulfur 34 in pyrite we find in the peritic coal. And this is because the oxidation of the sulfide mineral usually do not result in the sulfur 34 fractionation. So that means the sulfate originating from the pyrite is usually retaining the same fingerprint of the pyrite. Okay, so again, we find out that we have two sites where the origin of the sulfates, they may be assumed as to be just from pyrite oxidation without any other sulfate reduction, while again, both the two sites that in the slide before, they have an enriched value of oxygen 18, they have as well an enriched value of sulfur 34, which is again a proxy, let's say, it's a result, could be the result, sorry, of a sulfate reduction ongoing in aquifer. A thing to point out here is, so we measure as well the concentration of hydrogen sulfide, and for one site here, the Selene North, it's well, we have an high concentration of hydrogen sulfide in the outflow, but the sulfur 34 and the oxygen 18, they may be seen as just a pyrite oxidation as the only source of sulfate. Well, we can speculate that a chemical reaction basically between hydrogen sulfide and oxygen may actually mask the isotopic fingerprint of sulfate derived from sulfate reduction. That's because sulfate reduction actually deplete the hydrogen sulfide, and if we oxidize back that hydrogen sulfide into the sulfate, actually what we measure in the outflow could seem a fingerprint from pyrite oxidation, but actually in that aquifer maybe sulfate reduction is ongoing. So finally, to resume the important points, so we've seen that for our investigated site, a model carbon source could be an important source of alkalinity. And well, we propose that maybe sulfate reduction actually may enhance the alkalinity of the mine water, as this could result as a net alkalinity if that hydrogen sulfide in the product of this reaction do not get oxidized back as sulfate. Okay, and this is the same, or something the same process happened, for example, in anaerobic treatment systems. And also another important point is that about the interpretation of the sulfate isotopic signature, as it should take in account the potential reoxidation of hydrogen sulfide. Okay, and this reaction, for example, can happen when deeper coal mine water gets in contact with a more shallow water rich in oxygen. So hydrogen sulfide get oxidized back to sulfate, masking the real signature of the sulfate, which we could find in the deeper groundwater. Okay, so this actually, this is a process that may happen, and we may need to take in account to properly understand the deep geochemistry of the coal mine water. So last slide, which are then the implication for the heat schemes. So precipitation and oxidation of hydrogen sulfide, actually, they may mask the real concentration in the groundwater, because in the outflow, most of the time, we don't find really significant concentration, but deeper water, which is warmer, so actually we want to use that water for heating schemes, may be chemically aggressive, okay? So actually, this may be taken in account. As well, biogenic methane generation relies mostly on modern carbon source, okay, which is probably they are more bioavailable. So, and as well, for also in the first presentation, we have seen that there is a lot of wood actually in the mine working, and this is actually true. So we have a lot of leftover organic matters in the mine workings, which has a modern radiocarbon signature. And this organic matter that actually ferment, and that may be the reason why we have this methane with this radiocarbon content, pointing out a modern source. Or we can have a soil carbon dioxide infiltration. And the last point is about, as well, we find out that the microbiology is an important point in the geochemistry of the mine water. We have to bear in mind that the change in temperature of the groundwater may alter and may shape the microorganism community. And this actually can potentially increase the rate of both sulfate reduction and methanogenesis. So actually those processes, they may require a bit more investigation if there is a feedback in terms of methane or hydrogen sulfide when we inject, for example, warm water into the subsurface. Thank you very much.